which worked with FDA to develop iRISK. He'll be providing a brief live demonstration. Let me say a few words about the webinar logistics. You, the audience, will be muted, but the chat line is open. You can use the chat line to send us questions or let us know if you're having audio problems at any time during the webinar. We'll have several short question and answer spots throughout the program, and any questions or comments we can't get to today, we will address and post on the web, and we'll be sending you the link to that at some point. The purpose of this webinar is to introduce iRISK, FDA's new food safety risk assessment tool. As of this morning, iRISK will become available to the public and can be accessed at the URL you see on your screen. The webinar will provide you with an overview of iRISK and its purpose, what iRISK is, how it works, and who will use it. This will be followed by the live demonstration and a summary. Now I'd like to turn it over to Sherry for an overview of the purpose of iRISK.
Thank you, Susan. Before Dr. Chen presents on the specifics of iRISK, I have a few introductory slides. In its 2010 report, the National Academy of Sciences, or NAS for short, recommended that FDA develop tools for risk ranking in a risk-based system for enhancing food safety decision-making. And as you'll hear today, iRISK aspires to provide many of the attributes that the NAS identified for a good risk ranking model, that it should be fit for purpose, be scientifically credible, balanced, easy to use, and flexible. iRISK is an interactive, web-based system that enables users to conduct fully quantitative, fully probabilistic risk assessments of food safety hazards. So why iRISK? It provides several novel capacities. It enables the user to predict and compare risks and understand the effectiveness of different interventions relatively rapidly and efficiently. For example, iRISK structure allows comparisons across many dimensions, including different hazards, both chemical and microbial, different foods or categories of foods, and different production, processing, and handling events at any or all points in the food supply system, including the farm, processing, manufacturing, retail, or the home. It also allows comparison of different populations, such as elderly, pregnant women, or the immune-compromised. One reason iRISK can conduct risk assessments and evaluate interventions more rapidly is that it contains many built-in features, which Dr. Chen will cover in detail today. Another feature is that iRISK provides a straightforward user interface for ease of use, and likewise provides the results in a straightforward manner. iRISK also allows online access to ensure broad accessibility and allows users to save data and share it with others. So each time a user builds a scenario and obtains the results in iRISK, the data he or she enters builds the tool's knowledge base for future use, can be shared with others, and further extends its building capabilities. iRISK could not have been developed without the expertise and collaboration of many partners over the past 6 years. And as you can see from this slide, there's been continuous efforts to improve the capabilities of this tool. Emphasis has been placed on both the underlying structure and collection of data to populate a library of information to validate that the model is working as intended. And lastly, I want to mention that we are extremely grateful to the many experts that helped in the development of the product that we'll be making available today. Susan, do we have any comments or questions at this point?
Yes, we do. Someone is asking how FDA will use iRISK.
Yeah, that's a great question, and Yuan will address this in her presentation, so stay tuned for that.
And we have a second question. Is iRISK meant to replace other risk assessment tools for FDA?
No, it's designed to do certain things, but it will not replace all tools. It's merely one tool in our toolbox. As we'll hear about today, it's useful for exploring the many factors that influence public health risk. And I want to mention that in developing iRISK, we really wanted to enhance the capability to compare risk. And in doing so, we needed to balance the complexity of the mathematical equations with the need to have a tool that's flexible and can be responsive to current needs and events.
Thank you, Sherry. We'll move on now to Yuan, who will talk about what iRISK is and who will use it.
Well, thank you, Susan. My thanks also to the audience for joining the webinar today from the US and from around the world. In the next two sections, I will share some specifics about iRISK, its features, and potential applications. Through today's program, we hope to convey that iRISK is a tool that can estimate public health risk through objective, structure, and systematic integration of data and evidence. We'd like to highlight the capacity of iRISK for risk ranking, as Sherry mentioned, and at least as important, its potential to link preventive controls and interventions to public health protection. So what is iRISK and who will use it? As a risk ranking tool, iRISK can be used to compare the public health impacts of food hazard combinations in different ways. Here we have some examples. iRISK can compare one hazard in different foods, such as salmonella in fresh produce, shell eggs, and nuts. It can also help us compare multiple hazards in one food commodity, such as leafy greens that might be contaminated with enterohemorrhagic E. coli, norovirus, or cyclospora. And it can be used to compare multiple hazards in multiple foods. I will be talking more about the example of Listeria monocytogenes in soft cheese later to illustrate how iRISK works. Although the most obvious attribute of iRISK is the ability to compare risks across many dimensions, it can be used to do much more beyond risk ranking, which I hope will come to light in a more concrete way as we move along in the program. In the overview, Sherry mentions that iRISK development was a collaboration of experts. iRISK also built upon a well-established risk assessment paradigm using the Codex definition. The iRISK model followed this paradigm to integrate data from exposure assessment and dose response to characterize risk to the consumer. In fact, iRISK built upon risk assessment knowledge accumulated in the last 2 decades. For example, the understanding of contamination distribution in foods, dose-response relationships, and major process mechanisms in food production, processing, and handling. What can iRISK do? In a nutshell, iRISK enables users to construct risk scenarios more easily. Behind the scenes, calculations are carried out by iRISK using Monte Carlo simulation techniques. Once you've created a risk scenario, when you request a report, iRISK presents the results in 2 forms: a brief, convenient table that shows risk estimates and a ranked summary of the chosen scenarios, plus a printout of the full documentation of the data, assumptions, and references used in the development of the results. Later in the webinar, as Susan mentioned, our colleague from side will show a full report as part of the live demonstration. There are built-in features in iRISK that make the process of risk assessment streamlined and efficient. iRISK has a built-in model structure and a number of built-in mathematical equations and probabilistic functions. It also has a number of built-in templates for users to enter data that reflects their real-world or theoretical situations. By taking advantage of these built-in features, the user can focus on developing a process model that can include any or all stages in the food supply chain in a linear fashion. Evaluating the control measures and interventions of interest, and generally using iRISK to predict and compare public health impact for different risk scenarios. For the purpose of the webinar, we'll skip the built-in equations. And I suspect that for many in the audience today, you will be glad that we won't be getting into the equations at all, even though iRISK, not the user, actually does the math. The good news also is that the underlying model structure and math equations have been peer-reviewed by a panel of external experts. FDA conducted a peer review in late 2010. What you will see in the public release today, what we called iRISK 1.0, is the outcome of the first efforts since the peer review to improve and expand the capacity of iRISK as suggested by the peer review panel. Some of the examples of FDA response are enhanced navigation of the interface, more distribution templates, enhanced reporting, and I think that risk assessor, in particular in the audience, might appreciate this, enhanced model convergence and stability analysis behind the scenes. So now a preview of what iRISK can produce. The outputs of iRISK include risk estimates, risk ranking, and results on the impacts of interventions. First, for a single food hazard combination, risk estimates in several metrics are available, including the classic traditional total illnesses per year in the population, the mean risk of illness per serving, and also a health impact metric expressed as Deli or disability adjusted life years. So that's the output for each food hazard combination. Second, iRISK generates outputs that compare risk scenarios in a number of ways among a variety of food hazard combinations. Risk ranking currently by default is based on the Deli, but other metrics can also be used. Last, the outputs include results on the effects of chosen interventions at various points in the food chain. So who will use iRISK? We anticipate that iRISK will be useful for both technical and non-technical audiences. We think that decision makers in the government, industry, and other organizations will be important potential users of iRISK. Not to use it directly to develop models, but to put to practical use the results from iRISK, that is the public health metrics from risk models developed in iRISK. Risk assessors and food safety professionals in general will be the most likely users. They can use the tool to develop a risk scenario to help them understand and to represent in a simplified version the complex interactions of the food, the hazard, and the population, and to determine the impacts of preventive controls and interventions. We anticipate that iRISK will have users and audiences in academia, such as researchers, students, and the professors who educate the future of the food safety profession. Overall, iRISK can provide an efficient means for anyone around the world, really, who needs a platform to collaborate and to share risk scenarios. So with that, I'll stop for a minute and see if we could address any questions.
Thanks, Yuhan. We do have a question here that's very relevant to your last segment, and that is, will anyone be able to use iRISK, or do you have to have specialized knowledge and experience as a risk modeler? I am in public health policy. I am not a risk assessor. Will I be able to use iRISK?
I guess the short answer is yes. For the first part of the question, any user can use the tool either directly or indirectly. For the second part of the question, for someone in public health, you might not be doing the hands-on model building in iRISK, but as a risk manager, you may be in the best position to initiate the process because you would know the important public health questions that need to be answered and probably is in the best position to apply answers and results from the iRISK models. So we hope that iRISK will have useful applications for both technical and non-technical audiences. Right, any other questions?
That's it for that segment. And now we'll move on to a discussion of how iRISK works.
Okay, so how iRISK works, let's see. We have a diverse audience for the webinar today. So from our own experience, it's well known, I think based on our own experience, that risk is a term that is used in many contexts and is defined differently in different situations. So first and foremost, how does iRISK define risk? The endpoint in iRISK is the estimate of risk to the consumer, which is what we care the most about in food safety. This risk is defined as probability times consequence numerically. More specifically, it's the probability and extent of exposure times the probability and severity of consequence. So if you look at the left side of the slide, exposure itself is influenced by a number of activities at steps along the food supply chain, such as production, processing, transportation, storage, retail, cooking, consumption, and so on. Consequence, on the other hand, is affected by dose-response relationship for the hazard, probability of illness when exposure occurs, and population health effect. When the two sides merge together, we have risk estimate to the consumer. And the beauty of iRISK, we think, is that the tool is designed to do the merging for us. iRISK generates an estimate of risk to the population by integrating data and information on 7 elements. They are food, hazard, population, process model, consumption patterns, dose response, and health effects. I like to point out that the process model refers to the steps in the food supply chain, for example, steps in food production, processing, handling, up to the point of consumption. iRISK is flexible in that the user can enter data for each of the 7 elements and can also change the inputs to evaluate changes in the impact on risk given alternative data or assumptions, such as comparing with and without an intervention, comparing different interventions, different population susceptibilities, different pathogen strains that may have varying virulence, and so on. The data inputs can be point values, but most often will be probabilistic in nature, with a range of values represented by a distribution. Behind the scenes, iRISK integrates the data and generates results through Monte Carlo simulations. What we see in this figure is the relationship of the 7 elements of a risk scenario. These elements are interrelated. Each of them contributes an essential piece of data to create a complete risk scenario on which the estimate of risk is based. The fundamental question iRISK seeks to answer is a simple one. What risk does a food hazard combination pose to a population? The user needs to define the food, the hazard, and population of interest and enter evidence for the process model, consumption model, dose response, and health outcomes, such as a Deli template. And I will explain a little bit more about Deli later. So this is a screenshot of the iRISK interface that you will see more of in the live demonstration. The user enters data through this interface, develop models or view risk models also through the online interface from anywhere there is a connection to the internet. So as I mentioned, there is a built-in model structure behind the user interface. At a deeper level, in this figure, we see the inputs and the outputs of the model structure. The square boxes are user inputs, and the circular nodes are model outputs. Again, distributions are often used to describe variability in an element that reflects real-world situations, such as for contamination level, processing effects, dose response relationships, and consumption patterns. The inputs we see in this figure apply to a scenario in which the food is contaminated with a microbial hazard or a chemical hazard that causes acute effects. For chronic chemical exposures, the overall underlying model structure is similar. However, consumption patterns are defined differently, and the predicted result for the mean risk of illness is per serving, not for chronic chemicals, the predicted result for the mean risk of illness is per consumer, not per serving, as in the case for microbial hazards. For the rest of this section on how iRISK works, I will show a few examples to illustrate what users need to do to create a risk scenario. First of all, a complete risk scenario must include information and data for the 7 elements. Let's assume that the user has already defined 3 of the elements, have selected a food, hazard, and the population of interest. The examples I will share will cover the remaining 4 elements that are represented by quantitative data, that is, the process model, consumption patterns, dose-response relationship, and health outcomes. For the sake of simplicity, all the examples will be on microbial hazards. Because of its linkage to intervention in the food chain, I will elaborate a little bit more on the process model. And after I go over the user inputs in this example, in the live demonstration, our colleagues from RSI will show outputs from iRISK. What we see on this slide is a process model example for Listeria monocytogenes in soft cheese. The user inputs are the initial conditions, including Listeria contamination in soft cheese, and process steps in the process model that describes the effects of, in this case, handling on Listeria level at consumption. In this example, initial prevalence is defined by a single value, and the initial concentration is defined by a distribution, a triangular distribution on a log scale. By the way, the contamination data we see in this example came from a retail survey study in the US published in 2003. I would like to point out that there is a template also for the user to enter notes to document the reference, data source, and assumptions made in developing the process model. After entering data for the initial conditions, the user can develop a process model with multiple steps, describe the changes that can occur at those steps to the pathogen, and populate the model with data. In iRISK, there are a number of process types, and we'll see exactly what these are in the next slide. There are a number of process types that are built in to describe changes to the pathogen that can occur in production, processing, handling steps, where the contamination can change by growth, inactivation, environmental contamination, and so on. Because of the process model and the built-in templates for process types, iRISK provides a capacity to investigate the efficacy of different interventions and control measures across farm to table on the overall risk to the population, at least on a preliminary basis. So here are the 9 process types. There are 9 process types for the user to choose from to describe the changes to the pathogen at a process step. And they are increase by growth, increase by addition, decrease, pooling, partitioning, evaporation or dilution, redistribution, either partial or total, and no change because that could also be the outcome of a process step. The built-in process types provide flexibility in iRISK because these can be selected to represent the variety of common or major processes in food production, processing, and handling that affect the prevalence, the level, and spatial distribution of pathogens. The process model, as I said, is the means by which users can evaluate interventions at any steps along the food chain. So as a user, for example, as a risk manager, a user might be interested in using iRISK for nothing more than ranking and comparing the public health risk from different food hazard combinations. But beyond that, built in iRISK is the capacity to ask and get answers to what if kinds of questions. For example, what if we increase the heat treatment during processing of a low moisture food? What would be the effect, the predicted effect on contamination in the finished product and the number of illnesses? Another what if kind of question would be, what if the time temperature are improved for the storage of a product like soft cheese? What would be the predicted impact of the intervention on reducing the likelihood of illness? What would be the predicted effect on the total number of illnesses? And how would it differ for different population groups? There are intermediate outputs in iRISK, like what we see in this example for Listeria in soft cheese. And users can evaluate the effect of what-if control measures on the predicted contamination in finished product, for example, Listeria prevalence and levels in soft cheese. But perhaps of greater value, we think, is that iRISK enables the user to quantify the effect of intervention and control measure on the risk to the population. Therefore, iRISK has the potential to enable linking preventive controls and interventions not only to contamination in the finished product, which is very familiar, but also to public health protection. So that was an example for process model. Now let's take a look at an example for consumption patterns, soft cheese consumption by 3 different population groups. For each group, the user needs to enter data on consumption in terms of the number of eating occasions per year, that would be the number of servings per year, and the serving size. As expected, in the real-world situation, serving size is variable. In this example, it's a range of values represented by a triangular distribution. This data, the data that we see on this slide, came from a government survey and NHANES, the National Health and Nutrition Examination Survey in the US. Though I would like to point out that the data source for consumption, and for this matter, data sources for the other elements, can come from any country because this tool is not limited to any particular region. Again, iRISK provides templates for users to enter data and parameters. The next example I'd like to share is a Salmonella dose response model. In this case, a beta-Poisson model from the risk assessment literature. All right, advanced slide. Thank you. Yeah, again, this is an example on Salmonella dose response. When we plot this beta-Poisson model, plotted out as we would expect, it shows that the probability of adverse effect increases as the dose increases. The model just gives us the prediction of how it increases. iRISK offers choices of pre-structured dose response model templates, and the beta-Poisson model is one of the choices. The user selects one and populates it with parameters. Just as we can change the parameter in a process model, users can also change their inputs on consumption and dose response to conduct what-if scenario analysis, or to evaluate the uncertainty about the risk estimates. The built-in template for health outcomes in iRISK is a Deli template. Before I show an example, for those in the audience who may not be familiar, Deli is a commonly used health impact metric. It's well known that foodborne illness from different hazards may encompass diverse severity and duration. Some are short time and mild. Others may be serious and may require hospitalization. A small fraction of cases may lead to long-term chronic sequelae and even death in some cases. So to compare across this diversity of health outcomes requires a common scale, and Deli is one such common scale to use. This slide shows the Deli values for salmonellosis in the general population and listeriosis in the perinatal population in pregnant women. We can think of one Deli as one lost year of healthy life, a year loss of life. Based on current research and available data, a case of salmonellosis is translated into less than one Deli in this example. In comparison, the Deli value for listeriosis in the perinatal population is more than 10 because of multiple severe illnesses outcome, a longer duration, and much higher death rate. So the large difference between these two pathogens reflects differences in the disease burden. Again, iRISK provides templates for the user to enter a Deli value or compute the value from different health outcomes. Using a common metric such as Deli therefore helps to compare public health impacts from these two pathogens on a common scale. I think that concludes the section on how iRISK works. Before we go to the demo, let's see if there are any questions.
Thanks, Yuhan. We do have a question, and that is, can iRISK be used to predict contamination in finished products?
That's a good question. I guess the short answer is maybe, because it's kind of yes and no. iRISK can generate, and it does generate, intermediate results for contamination of a hazard, both in terms of prevalence and concentration in the finished product. That is the result from the process model, and this result is included in the detailed iRISK report. But right now, you will have to build a complete model, including all the 7 elements, in order to run a report and see the intermediate results. It's a good question. We'll need to think about how to include this capacity maybe in future versions.
Thanks, Yuhan. Another question. Can iRISK be used to compare pathogens and chemical hazards simultaneously?
Yes, you can use iRISK to compare microbial and chemical hazards in food at once. What you need to do is to set up the scenarios one by one for each food hazard combination, either a microbial hazard in food or chemical hazard in food. And once you create the scenario, then you can select some or all of the scenario for ranking, and the reports will show the ranking of scenarios that are built for a microbial hazard or for a chemical hazard. And I believe in the demo, we may see an example of that. Any other questions?
Yes, we do have a third question. Is iRISK linked to any database, such as NHANES consumption databases?
Right, so I mentioned NHANES in one of the example, but iRISK is not linked to any database. So the answer is not. It is not. Users can get data and use data from existing database, such as the NHANES database, and from literature and any other credible sources, but they do need to enter the data themselves when they create the scenario.
Thanks, Yuan. As promised, we will now move on to Todd Ruthman, who will provide a live demonstration of iRISK.
Control over to my desktop to do the presentation. Thank you, Yuan, for a very excellent and
don't
thorough
forget to hit the talk button as well, Todd. The lock button to broadcast your computer audio.
Just in full screen mode still? Okay. Yes, the audio should now be coming through. Yes. Okay. Requesting control.
Sorry, try it one more time. I'm sorry, try it one more time. There we go, great. There we go.
Excellent. Okay. Again.
Broadcasting your audio. You just need to flip back to your screen and we're good to go.
Excellent. Thank you. So again, thank you very much for Yuan's excellent presentation on the quite thorough and in-depth of how iRISK works. We have a short 10-minute window for this presentation, so my goal is to give you a quick look at how it actually looks online and to provide some hints and guidance on how to use the tool. So the first thing you're going to need to do to use the tool is to register. Our registration is currently closed, but will be opened up as soon as this webinar is complete. When you are required to register, just come to the home page of the website at iris.foodrisk.org and click on the register link. This will open up a page and we'll ask you for some small details. The most important elements on this page are a valid email address and your password. You need to provide a valid email address because we will be emailing you a verification email to activate your account. As well, you'll need that address if you need to reset your password later. The time zone is used to display on all the model pages. We show the last updated date, and we use the time zone information provided to show a date in your time zone. Once you have activated your account, you'll be told to go to the login page. You can click on login on the home page, or the top of the corner of the website also has a login button. Just type in your email address and the password that you supplied. You do have to click that you have reviewed the disclaimer, and then you can log in. As a new user, you will be be brought to the risk model page, which is where you will do most of the work in creating your models by creating hazards, foods, process models, and risk scenarios. But as a new user, you will have no information currently in your system, and so all the counts for each of the individual items are 0 and all the lists are empty. However, you can look at a read-only version of some sample models if you prefer to do this before using the tool. And you can go in and look, as in the earlier presentation, you can see how the Listeria data is entered into the system. We have all the dose response models created, all the health metrics created. We can go back and look at the soft ripened cheese foods and see how the consumption model has been created. and view all the population groups in there. So again, this is a read-only view of what's contained of some sample models. These sample models correspond to scenarios that we provided in a quick start tutorial. And we highly, highly recommend that all new users visit the help page when they first log in and download both the quick start tutorial and the user guide. They're both PDF documents, easy to read. And the quick start tutorial in particular, I won't go through in much detail here, but goes through page by page, step by step, with lots of screenshots, how to create all the elements required for 5 different risk scenarios on the microbial and the chemical side, and both on the acute and the chronic side, as well as how to run the reports and some guidance on interpreting the reports. So this is the key document. This is the place everybody should start when using iRISK. In fact, if you send us a support question, we'll probably ask you first thing, did you read the quick start? Once you have gone through the quick start, I'm going to log out of the empty account and I'm going to log into another account. Once you've completed your quick start, you will have something that looks a lot like this. You're going to have the same hazards and foods and process models that the sample model list has, except now that you can edit, delete, add, and make other modifications as you experiment and get more familiar with iRISK. As described in the earlier presentation, you have to define each of these elements in order to use the tool before you can run scenarios. Some quick tips about navigating around iRISK. The top of the page will just have your current account email address, ability to make modifications to your account, and a logout button. The top menu bar gives you access to the main sections of iRISK in terms of the model area, where you add all your information and review your data. The reports section, which you'll use to generate reports, as well as a sharing section where you can use it to share your models with other users in a read-only capacity. The sharing tool will be how you can coordinate and collaborate as a team by looking at each other's work. Sharing is described in the user guide, but not so much the quick start at this point. It's designed to be a fairly straightforward tool to navigate. It always use a consistent model of tabs across the top for the main sections, and then sub-tabs across the bottom for each individual element. Each page typically starts with a list where you can click on edit or delete. It will ask you to confirm a delete. So if we go in and click an edit button, I'm going to go back and click the Listeria node. Then you'll have sub-elements in here. So on the hazard side, you will go into each individual hazard to modify its dose response models, to modify the health metrics that are used, as well as quick links to any process models and scenarios that involve that hazard. As Yuan indicated in her presentation, you can also add as many notes as you need to any element in the tool. These notes are included to record your rationales and references and thoughts about each individual element, and they can be optionally included in the ranking reports. They are also shared with other people when you share a repository with them. However, you can indicate that some notes are private. If a note is indicated as private, then it will not show up in any reports and will never be shared with another user. To get back to the main page, you can click on the Models tab at the top or to My Models link in the breadcrumbs, and then you can go to each individual section. So in the food side, you can edit a food or add a new food and modify the consumption models, look at the population groups in the tool, and so on. Again, it's very consistent. You click on a tab, you get a list, you can edit or delete or add a new element. The add buttons, we won't go through them today because they're described in quite detail in the quick start, tend to be laid out like a wizard. So you'll be asked questions. For simple elements, you'll only be asked 1 or 2 questions. For more complex elements, such as process models, you may be led through 2 or 3 screens to fully define an element. Once you've gone through and you've created all your different elements, then you can create risk scenarios. So let's just take a quick look at an existing risk scenario. So here's a risk scenario for Listeria in soft cheese. You can see across the bottom that we provide quick links to relevant items in this scenario. So this one, you can go back to the food, the hazard, or the process model. You can change the name of the scenario and you can share it. So when you invite other people to view your models, only those scenarios that you marked as shared will be visible. However, when you do share a scenario, all the associated food, hazard, process model, and notes are automatically shared. You can go into the scenario and choose to include or exclude different population groups for different scenarios. So you may want to run one scenario where you're only looking at the perinatal population and then compare it to the total population. Let's quickly jump over to the report side. This is where you'll spend the other half of your time. There are two main reports available in this version of iRISK. The first is the model summary report. This is designed as an ad hoc report to give you a complete record of everything you've entered into your data. I'm just going to quickly generate. You can set different report titles and give an abstract if needed. You can pick and choose which elements are included. When you generate the report, take a second, it opens up a PDF document, and that PDF document reports back on everything that you've included in your repository. This doesn't do any calculations, this report. It's designed primarily for record keeping or for distributing to colleagues without providing access to your repository, or for doing offline reviews of data and information. It's a very useful tool, for example, if you want to assign tasks for reviewing dose response models to other team members. They don't need access to your models to do that. They can just mark up the document. The major report, though, the main feature for iRISK is the ranking report. When you look at the ranking report, you're going to be provided with a list of all your scenarios that you described, or any that have been shared with you through different repositories. Go back to my account. You'll also notice typically if you look at my account for an element, the toolbar will say My Models. But if you look at a shared element, it'll give you the name of the shared repository. Coming up on the end of our presentation, so I'm going to quickly do the report. You can filter based on these different elements if you have a long list. You can generate a report for everything that's currently listed, or you can pick and choose which elements you'll run. Grouping is described in the user guide, but it lets you combine elements. If you want to run two foods together or two hazards together, it will combine the total DALYs in the ranking. At the bottom of the page will be a list of any incomplete models. So this will try and give some guidance of anything that might be missing in your scenarios. For example, you might not have added the population groups, or you might have put a health metric of 0, which will return 0 risk, and so that's not permitted. Different elements, we try to provide some guidance as to what's wrong. You click the Generate Report, the page will reload, and you'll be told that the scenarios have been submitted, and to go to the report history to check progress. We have a queuing system set up given the number of users we're expecting, and so it's first in, first out in terms of the models being run. When you come to this page, if your model has not yet been executed, it'll be appearing in the pending section. It will either say in process, which means it's running right now, or it'll tell you where you are in the queue. You can just check back periodically, or you can submit 3 or 4 jobs and then come back a bit later and log in. If you're doing it real time, you can just hit the refresh button and it'll flip down to the completed reports at the end when it's done. By default, we show you the last 2 weeks worth of reports, but you can change this to the last week or all, or different time frames. To view a report, you basically just click the View Report button, and you can optionally include details and notes. And you want to show the first couple pages of this report, but this report has the title and any abstract you might have put in, and then gives you a ranking based on the health metrics that you selected. If you've used grouping, then multiple scenarios will appear under each group. If you include details, you will see all the details and all the intermediate results in the following pages. And if your scenario has notes and you include notes, they will also show up in this report. So this report is useful for distribution to, again, for colleagues, for record keeping, and for other purposes. You can come back. We store the results of all your runs. So you can come back at any time and reprint that report, but it will always use the most current notes. This is to allow the mechanism where you may have found some typos in some notes and you just want to quickly change them and reprint the report. You don't need to rerun the ranking to do so. If you find you've run a lot of reports you don't want, you can click on any report and delete it, and it will be removed from your list. I think we're running a little over on time, so I'm going to wrap it up there. I think the main takeaway from this is once you register, please read the quick start and follow the steps involved there to fully understand how the tool comes together. As you may have gathered from the previous presentation, while we designed this tool to be easy to use, you do need to understand how it works as a whole in order to effectively use the tool. At this point, I think I'll hand it back to GIFSAN FDA to let me know if there's any questions that have come up.
Thanks, Todd, for that very informative walkthrough. I have received 2 sets of questions for you, and I'll give you the first cluster together since they're all related. When can I create an account in iRISK? Are there any requirements for creating an account? Is iRISK free? Do I need to buy any software to use
Okay,
iRISK?
that's a bunch of different questions. First one, registration is going to be open in about 5 or 10 minutes as soon as this webinar is closed. So please feel free to register. There is no cost associated with registering or using iRISK as a software package. It is a web application, so you do need to have access to the web and obviously to use it. You will require a PDF reader, but that's fairly standard. As well, because it's a web application, the user experience is going to vary a bit depending on which browser version and software you use, but by and large we've tried to design it to be usable by a wide range of software and browser systems. So to reiterate, as soon as we all hang up on the call, you can go register for iRISK.
Thanks. Likewise, the next 2 questions are related to each other, so I'll ask them together. How much training does it take to use iRISK, and is in-depth training available?
Okay. I think you probably could gauge from the presentation how much training you feel you would need to use the tool. It does depend a great deal on your individual experience and what you want to use the tool for. If you're a risk assessor, very familiar with food risk concepts and doing risk assessments, then you should be fairly easily to use the tool once you run through the quick start. If you're a risk manager, you may need some guidance on interpreting the results that are coming out of the tool. We are working on additional training materials and so on, which will be posted. We'll also be updating the user guide and quick start as we get feedback. But some people may require additional training, particularly around appropriate parameterization of their models and interpreting the results.
Thanks again, Todd. Before we move on to the webinar summary, I have another question for you on here. Is there a way to protect confidentiality of the data? If I enter my data online, does FDA have access to it?
Thanks, Susan. I could take that question. The tool, as you've seen in the webinar announcement,
It's going to be hosted by GFN. FDA does not have access to the data. The site is protected by password, and only the user would have the password to their account. It's foreseeable that in order to, in some diagnostic situation, troubleshooting situation, the web manager might need to access
need to
the data,
access
and
the data.
this would be upon request
This would be
by
upon
the
request
user and with
by the
permission
user and
by
with
the
permission
user to
by the
access
user
the
to
data.
access the data.
Oh.
Can you mute your speaker for us real quick?
Oh, okay.
Oh, okay.
Okay. Okay. Thanks, Tom. Thanks, Tom.
We can...
Yes, sorry.
Okay, so
having echo on that. We need to stop that so users can hear.
right. So I guess in certain troubleshooting and diagnostic situation, the web manager, upon the request of the user with permission, could access the data for technical support reasons, but FDA does not have access to the data. We recognize that this is an important issue, so we're looking into additional protection features. And anything else, Susan?
No, we can move on to the summary now, Yuan.
Okay. So in summary, iRISK is one of the latest developments in FDA's growing risk portfolio. We've used iRISK in a number of ways. In the past several years, starting with a beta version of iRISK, we've commissioned several studies to develop a library of risk scenarios for food hazard combinations of interest, and this work is ongoing. We've used the models in iRISK to evaluate the effectiveness of interventions such as on-farm interventions, control measures implemented in processing, and changes in retail or consumer handling practices. In developing iRISK, the tool has served as a means for us to collaborate with other government agencies. iRISK is also a useful platform for us to more effectively communicate risk assessments to decision makers and to share data and risk scenarios with our collaborators. In the future, we anticipate a broader use and a greater degree of application of iRISK to support risk-informed decisions. In order to reach the full potential of iRISK, though, in particular to overcome some of the data gaps and limitations, we do recognize that we need to collaborate and leverage resources. This includes government, industry, and academia collaborations. We also need to articulate key risk management questions to answer. That means to keep the dialogues open and continue to have more interactions with our decision makers. And this is particularly important as we move forward so that the right scenarios are developed, validated, and deployed. And we need to work together on targeted data collection, including prevalence and enumeration data in the food supply chain, so that we can better understand risk associated with normal baseline situation compared to, for example, an outbreak situation. Our vision for iRISK is a web-based system where a community of stakeholders contribute data, model, and intervention information in a manner that is consistent, documented, systematic, structured, and quantitative, all of which hopefully will lead to an improved understanding of risk in the food supply system. iRISK, we think, can function as a comprehensive repository for data derived from credible sources and up-to-date literature, and to which each user may choose to contribute his or her data entries and results, so that as to enable future users to use them or build upon them. This in turn would hopefully fuel the capacity of iRISK for enabling risk assessment and ranking, comparing intervention options, and accumulating knowledge to enable rapid predictions. So to conclude, as Sherry mentioned at the very beginning, we want to take this opportunity to acknowledge the many experts who have helped develop iRISK and really offered their critiques to improve it. In particular, IFT, the Institute of Food Technologists, and the expert panel they assembled to develop a prototype on which iRISK evolved. Our colleagues at Risk Sciences International, RTI International, and the external peer reviewers. We also want to thank colleagues from a number of agencies, both in the US and abroad, organizations and universities who participated in several beta testing and preview, and for providing their thoughtful comments to help improve the system. For more information, we encourage you to visit the foodrisk.org and the FDA risk assessment websites. iRISK 1.0 is the first public release. We plan to collect feedback from users to further enhance the tool where necessary. With that, I'd like to turn it over to Susan for closing remarks.
That concludes today's presentation, and a recording of this webinar will be posted at the foodrisk.org website within 10 days. And with that, I thank our speakers, and I thank you for your participation.